Hello, this is Haley Weber with Gabriella Anderson, Gracie Wright, and Natalia Benitez Hilton. I hope I got that right. And we are going to be talking about leprosy.
Prior to 1873, the cause of leprosy was thought to be some form of sin or uncleanliness. Leprosy was thought to be a form of punishment to those that had sinned. Priests were instructed by the book of Leviticus in the New Testament to cast those with leprosy from society and label them unclean. A person with leprosy would be given the same religious rights as someone that was dying, creating the stigma that those with leprosy were the walking dead. Medieval law stated those with leprosy were to be stripped of their citizenship rights and property. They were then to be left on the streets to beg so that communities could be reminded of what God's punishment looked like. People with leprosy were thought to be able to spread the punishment through contact alone, causing people to attempt to isolate those with leprosy. Eventually, the term lepers was created as a form of persecution and ostracism. In 1873, Norwegian physician Gerard Hansen discovered the causative agent of leprosy was Microbacterium leprae while working at the Leprosy Research Center, St. Jorgen Hospital, founded in 1400 AD in Norway due to the commonality of leprosy. This discovery directly contradicted the idea that leprosy was the result of sin and that it was easily communicable. Hansen delayed publishing his discovery because he was unable to inoculate M. leprae into animals. Eventually, Hansen attempted to inoculate a human patient without her consent. Despite criticism for his unethical research, he gained recognition in 1897 for his discovery that leprosy was a non-fatal chronic infectious disease. This eventually led to leprosy being renamed Hansen's disease. In 2008, by analyzing the liver of leprosy patients from Mexico and Costa Rica, Dr. Han and Dr. Silva found significant genetic differences between M. leprae and the microbacterium found in their livers. Their study concluded another causative agent of leprosy was M. lepromatosis. Up until 2005, leprosy was believed to have originated in India. Recent studies into historical text, genetic, and skeletal data has altered that belief. Historical texts can be quite hard to interpret due to translational errors and/or misleading and non-descriptive terms, but they are still able to provide some insight into the history of leprosy. The Sushruta Samhita is an ancient medical and surgery text from India. It described an illness, kushtha, that translates to eating away, and all its forms. Stating in detail the symptoms now associated with leprosy has led to the belief that leprosy was in India in 600 BCE. The Nei Qing is one of the most ancient medical texts from China. This and other medical texts from China describe oqi, or leprosy, cases in detail and call it an evil disease. These texts put leprosy in China in 2600 BCE. Greek authors such as Lucretius and Plutarch spent their lives traveling and sometimes documenting what appears to be cases of leprosy. Lucretius states leprosy was only found in Egypt, while Plutarch stated leprosy originated in Greece. These texts place leprosy in Egypt and/or Greece in 100 BCE to 40 BCE. Though the Bible in its current state mentions leprosy, it is not the leprosy we see today. The term leprosy used in the Bible was denoted as unclean and was used to describe a variety of skin conditions such as boils, eczema, scabies, and more. This prevents the Bible from being a reputable source for determining leprosy's origin. In 2005, a study used comparative genomics to discover and track 4 strains of M. leprae. Strain 1 was found in East Africa, Asia, and the Pacific region. Strain 2 in Ethiopia, Malawi, Nepal, North India, and New Caledonia. Strain 3 in Europe, North Africa, and the Americas, and strain 4 in West Africa and the Caribbean. The study concluded that due to how M. leprae strains appeared to spread along migration, colonization, and slave trade routes taken from East Africa, it must have originated there 100,000 years ago. DNA from skeletons has found evidence of leprosy, like skin lesions, and sometimes M. leprae in India, Europe, and Turkey around 2000 BC. A more recent study was able to find 10 ancient genomes from M. leprae in European skeletons dating back to 400 CE. This study believes that they will be able to find more cases of these 10 genomes, or even more ancient genomes of M. leprae, and be able to prove that strains of leprosy were found in Europe early enough to put Europe as the origin country of leprosy. The combination of historical, geographical, and skeletal data collected in hopes of determining the origin of M. leprae is lacking and sometimes contradictory. In addition, the only confirmed hosts of leprosy are 9-banded armadillos in the US and red squirrels in the British Isles. All of this information confirms that the origin of leprosy is not somewhere tropical, despite most cases today being in tropical areas.
The symptoms of leprosy can lay dormant for upwards of 20 years before manifesting. Symptoms typically include skin lesions. Without treatment, leprosy can cause nerve damage that oftentimes results in long-term disability and deformity. Um, it can cause anesthesia on the soles of the feet as well as the facial nerves. Blindness has also been associated with leprosy. When left untreated, it will result in thickened skin and leonine facies, which is the picture on the side. And the hair on affected parts of the skin will start to fall out, especially on eyelashes and eyebrows. Earlobes will start to thicken, as well as the skin on the legs, and the leg skin can also become dry and scaly. The exact mode of transmission is not completely known or fully understood, but what we do know is that it cannot be transmitted through touch. So if someone touches you with leprosy, you will not get it that way, but because it can't infiltrate into or out of the skin. Individuals and families with leprosy or people in communities with a high amount of leprosy are more likely to contract the infection. Transmission likely would happen when someone infected with leprosy sneezes or coughs or somehow puts droplets into the air that can be picked up by someone else's respiratory system, and then they become infected. Studies also found that soil can be contaminated with an infected person's spit, mucus, and bathwater, and that could be potentially infecting other people.
In general, leprosy is low in virulence, and the majority of people who contract it and receive treatment do not die. But there are two main strains of the microbacterium that cause leprosy. M. leprae, which has low virulence and is the more common strain, and then M. lepromatosis, which is of higher virulence and causes diffuse lepromatose leprosy, or DLL, which is the way easier way to say it. And it's more of the rare strain. So Microbacterium leprae, M. leprae, is thought to have been around for approximately the last 100,000 years, at least in people. Whereas Microbacterium lepromatosis was discovered in 2008 in the bodies of patients who had died in Mexico and Costa Rica. This is the more virulent strain as it causes more deaths and more severe symptoms than M. leprae. In this case, the DLL is essentially when the skin becomes so tough and thick that like wrinkles in the skin disappear. And so sometimes it's called the pretty leprosy because it makes you shiny and smooth. And it is considered to be very rare. In a phylogenetic survey of 227 samples of tissue from leprosy patients from South America, only 6 of the samples they found contain M. lepromatosis. So there is a little bit of evidence that suggests that M. leprae and M. lepromatosis are on the path to becoming at least slightly more virulent. So first, leprosy is spreading across the globe, including the United States. In the last few years, there have been a rise in leprosy cases in the eastern United States that were not linked to contact with armadillos, which are the known carrier of M. leprae in the US that are linked to past outbreaks. Thus, it is kind of an emerging infectious disease in the US, at least in some parts. There have also been a rise in cases of M. lepromatosis in South America and parts of Asia, though this one is still very rare. But this does suggest that virulence may be on the path to increasing Another fact that may suggest that virulence is evolving is that M. leprae is able to live in free-living amoeba for up to 8 months and maintain its virulence. So this was demonstrated in a study where researchers injected amoeba that had M. leprae in it into a sample of mice after it had been in the amoeba for a few months. Mice that were given the injection developed lesions in the and infected the mice that were not given injections. So this suggests that leprosy can maintain its virulence in non-mammalian or reptilian hosts. While this doesn't exactly suggest that leprosy may be becoming more virulent, it is still worth thinking about and may play a role. That being said, there's not enough data to justify panicking yet. There's still much that remains unknown. All of these findings are preliminary. And our genetics honestly might just be a better indicator of how virulent a virus is to us, this virus is to us.
So, what do we not know about leprosy? It has been shown that we do not know the mode of transmission of leprosy. As discussed earlier, mode of transmission is one of the biggest unknowns when it comes to leprosy. They have discovered that touch is incapable of being a mode of transmission. Researchers have made experiments and found results, but not enough to prove the modes. Currently, most people think that it is spread through a respiratory route. This experiment will help assist with understanding the assumed modes of transmission of leprosy. By using different modes of transmission to put Mycobacterium leprae in contact with live lungs, which mode would represent the most tracing of the bacterium? We hypothesize that if the Mycobacterium leprae is put in contact to a lung through injection of human saliva, it will create the most tracing of the bacterium. This experiment would need to contain 4 lungs that are being kept pumping by recent technologies. As humans and 9-banded armadillos are proven to be carriers of leprosy, they will be the 2 organisms we pull the Mycobacterium leprae from. The lungs need to be clean of all Mycobacterium leprae before the experiment begins. The 4 modes of transmission being used will be a spray replicating a human sneeze that contains Mycobacterium leprae, a spray replicating the sneeze of a 9-banded armadillo containing Mycobacterium leprae, injection of human saliva containing the Mycobacterium leprae, and injection of 9-banded armadillo saliva containing Mycobacterium leprae. We do know that touch is not a mode of transmission of leprosy, which is why we left that most obvious mode out of the experiment. All of the modes of transmission will contain the exact same of Mycobacterium leprae. They will be placed on and into the lungs on the first day of each month. Because this disease is chronic, the experiment will take exactly a year. These lungs will be monitored daily, and once a year has passed, they will be tested to see which lung contains the most Mycobacterium leprae. For the hypothesis of this experiment to be correct, the lung that was injected with the human saliva would need to be the lung that is tracing the most Mycobacterium leprae at the end of the year. This experiment can help designate which assumed mode of transmission produces the most Mycobacterium leprae in a human lung over a year. This could assist in the prevention of the disease in the future and further research on the mystery of how this disease spreads. Thank you.